Since microbiota can influence our health, so what matters? Quality, quantity, or activity of microbiomes. Over the last decade, pioneering papers have shown that some metabolic disorders such as obesity and diabetes are associated with shifts in the microbiota at the phylum level. Since this discovery, not all papers have been able to replicate this finding, which gives rise to the following question: should we focus only on the general composition at the phylum level, or should we go deeper? However, in addition to these considerations, there is another important question. Is it more relevant to explore the metabolic capacity of the intestinal microbiota and eventually to the metabolites produced than it is to study the microbial composition alone? At this stage of our knowledge, there are no clear answers to these questions. Along these lines, in addition to these discoveries and observations, a recent very elegant study demonstrated that it is the absolute quantity of microbiomes and not the proportions of microbiomes that really matters. This study strongly argues that most previous studies, which have primarily been based on the investigation of the relative properties of microbiomes, have probably not been able to capture the essence of the problem. For example, the authors observed differences of up to 1 log between the microbial loads of healthy individuals. As another striking example in this paper, the authors highlight the fact that the abundance of bacteria is associated with Crohn's disease only when used the classical relative abundance-based profiling, whereas by using quantitative microbiome profiling, it was the abundance of the genus Provitilla that was found to be decreased in patients with Crohn's disease. Moreover, this observation analyzed the real risk and the limitations of using relative abundance-based analysis, because this type of analysis can lead to enormous interpretations. Similarly, the necessity for analyzing not only the presence of certain gut microorganisms, but also the activity of these organisms has already been discussed by authors. More recently, Schirmer profiled the gut microbiomes of more than 100 individuals, including patients with Crohn's disease, ulcerative colitis, and non-IBD control patients, and analyzed both the metagenomes and the metatranscriptomes of these individuals. They discovered that most of the bacteria were well correlated when analyzing DNA and RNA abundance. However, strikingly, some organisms that were abundant in terms of the metagenomic data were, as stated by the researchers, inactive or dominant in the gut with little or no expression. Conversely, some disease-specific bacterial characteristics were only detectable when analyzed at the transcriptional level. Therefore, such findings highlight the fact that the transcription of specific gut microbiome pathways may vary over time, leading to potential phenotype changes that may be complementary to changes associated with classical metagenomic abundance. Having said that, the specific examples highlighted above and relating the quantity of bacteria and the activity of bacteria strongly demonstrate that the importance of multi-omics approaches and how various genomic and metabolomics approaches complement each other to further dissect the interactions between microbiomes, host, and overall metabolites. Several interesting studies have also proposed that the microbiome should be seen as an integral part of a precision medicine approach because it contributes to the inter-individual variability in all aspects of disease situation, but is also a modifiable factor leading to the development of future therapeutics. Let's go to the topic of gut microbiota and obesity. The first evidence on the role of the gut microbiota on host adiposity came from studies in germ-free animals. Germ-free animals mean that animals devoid of bacteria and bred in sterile isolators. In 1983, Wostman and colleagues observed that GF rodents require 30% more calories to maintain their body mass than conventional ones. Jeffrey Gordon's laboratory at Washington University first found that conventionally raised mice have 42% more total body fat and 14.7% more continental fat than GF mice, although GF mice consumed more food. They pioneered the investigation of gut microbiota as a factor influencing fat storage and obesity. They next demonstrated that colonization of GF mice with a circum-derived microbiota from conventional mice produced a 16% increase in body fat mass with 2 weeks. The increase in body fat was accompanied by insulin resistance, adipocyte hypertrophy, and increased levels of circulating leptin and glucose. This was partly explained by the capacity of the gut microbiota to degrade undigestible polysaccharides into monosaccharides, which could be absorbed, leading to increased hepatic lipogenesis in the host. Moreover, inoculation of gut microbiota suppresses the ANGPTL4, a circulating inhibitor of lipoprotein lipase. Conventionalization leads, therefore, to increased adipocyte LPL activity and then to increased cellular uptake of fatty acid and adipocyte TJ accumulation. The physiologic importance of ANGPTL4 was further established by the demonstration of GF. ANGPTL4 deletion mice have the same degree of adiposity as their conventional counterparts. Finally, in contrast to conventional mice, GF mice fed a high-fat, sugar-rich diet are protected from diet-induced obesity. This lean phenotype was associated with increased skeletal, muscle, and liver levels of phosphorylated AMP-activated protein kinase (AMPK). AMPK is an enzyme that is conserved from yeast to humans and functions as a fuel gauge that monitors cellular energy status and stimulates fatty acid oxidation in peripheral tissue. Hence, the gut microbiota may ease skeletal muscle fatty acid oxidation through a metabolic pathway involving phosphorylation of AMPK. It was further observed that GF mice receiving a high-fat diet showed enhanced insulin sensitivity with improved glucose tolerance and reduced insulinemia in comparison to conventional mice. So, the role of bacterial fermentation in host energy harvest and appetite regulation. Important mechanism that can explain differences in body fat between conventional and GF mice is the increase in energy harvest from the food due to the fermentation of the gut microbiota. The gut microbiota is able to process complex dietary plant polysaccharides otherwise inaccessible to humans, to monosaccharides and short-chain fatty acid, principally acetate, propionate, and butyrate. This short-chain fatty acid represent an important energy source for our body as they can provide approximately 10% of the daily energy supply in omnivores and up to 80% in herbivores. Beta-rich is a preferred source of energy for colonic epithelial cells and increases the density of capillaries analyzing the small intestine various epithelium. Absorbed propionate and acetate are delivered to hepatocytes where they can be used for glucogenesis and lipogenesis respectively. However, short-chain fatty acids act not only as energy substrate for the host but also as signaling molecules influencing energy intake and metabolism. As we mentioned previously, short-chain fatty acids are ligands for at least two GPR receptors. These GPRs are mainly expressed by gut epithelial cells, in particular into endocrine cells. FFAR2 is preferentially activated by acetate, and FFAR3 by beta-rich, whereas propionate activates both receptors. FFAR2 deletion mice was found to resist to diet-induced obesity, indicating that FFAR2 could promote fat storage. Similarly, it has been shown that GF-FFAR3 deletion mice colonized by a microbiota gained less fat mass than their wild-type lemurs. The authors proposed that in the absence of FFAR3 signaling, the plasma levels of peptide YY is reduced, leading to an increased gut motility and reduced energy harvest from the diet. Therefore, the FFAR2 and FAR3 would be modulators of host energy balance through efforts depend on the gut microbiota. These receptors may also control eating behavior as increased production of short-chain fatty acids due to fiber administration leads to increased satiety and reduced food intake. Short-chain fatty acid produced by the microbiota may constitute a fine-tuning of the host metabolism by the regulation of energy harvest, fat storage, and appetite. Okay, that's all. Thank you.